this lecture, all we're going to do is start examining the first of the major biomolecules, proteins. Remember, there are several biomolecules: proteins, carbohydrates or sugars, nucleic acids, and lipids, which would be fatty acids and things like cholesterol. So in this lecture, we're going to start with proteins. Before we begin, let's take a look at what we've already seen a little bit from previous lectures. For example, we've seen that eukaryotes and prokaryotes actually use membranes to isolate the cell from potentially harmful environments. So to have a perpetuating chemical reactor called the cell, we have to isolate it from the environment, otherwise the chemical reactions will simply diffuse away into the environment. We could not have a self-replicating or self-continuing system. The eukaryotes go a little bit further, and they have optimized certain dangerous chemical reactions for very specific environmental conditions, such as cellular digestion taking place in the lysosome. So what this means is the eukaryotes have created compartments for specific metabolic processes. And you should be familiar with those compartments, things like the lysosome, the cytosol, the mitochondria, the endoplasmic reticulum, and we will see that metabolic events can move between compartments. For example, there's aspects of fatty acid synthesis that will take place in the smooth ER as well as the cytosol. But usually we need to keep very specific metabolic systems away from each other. We also learned that cholesterol esters and fatty acids are broken down, or catabolism is performed in the lysosome. This is very important for cellular health, as you have to have cholesterol within the cell membranes, and that we also tied failures of specific enzymes, cholesterol ester, hydrolase, and lysosomal acid, fatty acid lyase, to a couple of diseases, including Wolman's disease, which is an astoundingly dangerous disease, and cholesterol ester storage disease. We have seen already that protein functions are driven by the weak interactions, things such as hydrogen bonds, van der Waals interactions, hydrophobic interactions, electrostatic interactions. We've seen the weak bonds can be affected by changes in pH or the abundance of proton, so that if we have negative charges on a protein, overabundance of proton could lead to neutralization of those charges, which would in turn affect the ability of those amino acids to form weak bonds. And we're going to see that a little bit more here. Buffering, we very briefly touched on. It's not a major concept that we're covering, and compensatory respiratory alkalosis is also not a concept we're covering heavily, but it is related to buffering. It's how your blood pH maintains a steady 7.4. So now we're going to just talk about proteins, their compositions and structures. From Tomasco, this is chapters 3 and 4, mostly chapter 4 and a little bit of chapter 3. So your goals: 1. To understand the different protein functions. Like all biomolecules, proteins have more than one purpose, so we'll be covering those. You are to learn the amino acids, their names, their categorization, their 3-letter abbreviation, and their single-letter abbreviation. And while I don't require students in this course to memorize the amino acid structures, you would find it very helpful to memorize them to further understand how proteins and especially enzymes function. We will also look at the different levels of protein structure. Here we're talking about primary, secondary, tertiary, and quaternary, and how to distinguish between them. We'll look very briefly at protein folding and protein motion, and then we're going to talk about a very unusual disease, the prion disease. So our key concept for functional roles of proteins is that proteins have different dynamic and structural functions. There are different things that they do. We have sort of a little overview here of the dynamic idea, the chemical reactive enzyme. So an enzyme is just a type of protein, and so an enzyme will recognize a substrate. The substrate will bind to the enzyme, chemical reaction will take place, and the products are released. Now, while this schematic diagram really oversimplifies things, the idea is sound, that enzymes find their substrates and convert them to products. We also have structural concepts behind proteins. So these we can see in this structure here. This is a tertiary structure overall, which we'll cover why that is soon. And we have these green arrows are beta sheets, and these purple cylinders are alpha helices. And there are these gray random coils. Now bear in mind, this is a crystal structure, and no protein looks like its crystal structure. The crystal structure of a protein is its average structure under completely non-biological conditions. But usually we see that that structure is at least a reasonable approximation of how the protein could kind of look under biological conditions. So what functions do proteins have? Well, the first is structural. Proteins actually very frequently act as scaffolding and support for a cell or for an organism. So in us, proteins form the matrix for bone deposition. Proteins are connective tissue. And collagen, for example, collagen and elastin, giving tensile strength and flexibility, all done by proteins. Dynamic functions. Proteins have dynamic functions. For example, catalysis of metabolic reactions. This is the domain of enzymes and will be the primary focus of this course overall. There are thousands of unique metabolic actions, and almost each one is going to require a specific enzyme. Under dynamic, we can also have transport, so proteins like hemoglobin and myoglobin allow for the transport of oxygen in the blood and muscle respectively, and we have some additional dynamic functions. Here's contraction. Myosin and actin allow for contraction and changes in cellular shape. We can also have motor proteins, we have turbines, we have all kinds of dynamic functions. We also will see proteins performing protective functions. Immunoglobulin and interferon can bind to foreign substances or direct natural killer cells to foreign cells or to foreign substances for elimination. The protein fibrin appears during an injury to stop loss of blood. We also have control functions for proteins. Hormones, for example. These are fairly simple proteins. They don't have catalytic activity. For example, insulin, it's just a protein. It's involved in sugar homeostasis. It will bind to a receptor on the target cell, allowing the target cell to take in sugar and clear it. Somatotropin is a very simple protein that has a whole host of effects on different cells. This is the growth hormones. You can imagine organisms as they're growing undergo tremendous numbers of organismal changes driven by somatotropin. More simply, we can have control and regulation of gene expression. Transcription factors such as NF-kappa B, P53, P21, histones, which help organize DNA, and polymerases. Now these are enzymes, but they help replicate DNA and make RNA. Just for virtue of looking at that for a moment, this picture is illustrating the central dogma of information transfer, and that we have this idea that we have a gene that contains the codons for amino acids. That gene is transcribed into an mRNA sequence that is then ready for translation by a ribosome to give you a specific protein. The problem with the central dogma is that it implies that the flow of information is unidirectional, and it isn't. Proteins will play a role in absolutely every aspect of this. So proteins can control mRNA translation, proteins will control transcription, and mRNA itself can control transcription and translation. So it's not a unidirectional flow. It's more of an extraordinarily complex web of information transfer. So moving on to what makes up proteins, well, those would be the amino acids. Now, there are 20 common amino acids that are used in the genetic code. There's a handful of others, particularly in bacteria. They'll use some strange things like selenomethionine, but we're really focusing on the 20 common. Now, amino acids are all basically the same, except for what's called their side chain or their R group. This is what's going to determine their characteristics. Amino acids have particular characteristics including handedness, ionization, volume occupied, potential to form different types of bonds such as hydrogen bonds, ionic bonds, or disulfide bonds. So it's important to remember that amino acids are chiral with the exception of glycine. Glycine is not chiral, and there can be L and D isomers. This is different from R and S. Metabolism and biochemistry by extension don't generally use the RNS designator. We use L and D. There is no formal relationship between L and D. L means levorotatory, D means dextrorotatory, and it talks about how these molecules rotate light when they're in solution. It has nothing to do with the RNS naming conventions, which are a completely different thing. For us and for all of life, we use the L isomer, but the D isomer does appear, and it is the job of the peroxisome to eliminate D amino acids. As a note, the peroxisomes do a few other things: long-chain fatty acid oxidation, lipid oxidation, oxygen-based reactions, and some peroxide-based reactions. But we're focusing here that the peroxisome eliminates D amino acids, leaving the L amino acids for use as the building blocks of proteins. This is our generic structure of an amino acid. The ionization is actually correct for a pH of about 7. We have the amine on one end, the alpha carbon, a carboxylate on the other end, and a side chain. And this side chain is what varies between all 20, with the exception of glycine, where this will be hydrogen. I've mentioned disulfide bonds a couple of times. The only amino acid that makes disulfide bonds is cysteine. Methionine cannot do it because the sulfur is capped by a methyl group. If cysteine undergoes oxidation, so these hydrogens are removed as protons along with an electron, you get the disulfide bond. Under reducing activity or a reducing environment, that disulfide bond can be broken to two independent thiols. This is a really important reaction for the protection of the cell from oxidative damage, and is found in the molecule glutathione, and we're going to cover that a little bit later on. Just to show, a disulfide bond can appear within a peptide chain or between peptide chains, so within or between. And this is going to be one of the few, if not only, covalent bonds we will see outside of the primary sequence covalent bonds. So these covalent bonds help promote tertiary and quaternary structures, which will make more sense when we get there. It should be noted that peptide bonds are not flexible. Not shown on this nitrogen. There is a lone pair of electrons on this nitrogen, which can then form an additional double bond between the carbon and the nitrogen, allowing the electrons of this double bond to relocalize to the oxygen. This creates a resonance structure within the peptide, and the peptide actually adopts a planar configuration and does not have rotation. Don't confuse that with these bonds between the carbons. These bonds have rotation. It's the peptide bond, which forms between the nitrogen, the carbon, and the oxygen. We need all three of these in this arrangement to create the peptide bond, in which these two bonds, the one between the nitrogen and the carbon, and then the carbon and the oxygen, all possess double bond character. And just to emphasize a little bit more, I'm showing you here the double bond character within the peptide system and the single bond rotation. Phi and psi have single bond rotation. They're sigma bonds, no problem. And this peptide bond here does not have rotation. This is a flat planar bond. And so what are the 20 amino acids? Well, they're shown here, and you're responsible for all 20 of them. There are 10 classified as nonpolar, though there's some arguments between cysteine and tryptophan as to whether or not those are actually nonpolar, but they tend to be hydrophobic, so we'll go ahead and class them as nonpolar. Cysteine is very unique in that this is the amino acid that forms disulfide bonds. So you have 10 nonpolar amino acids. Of note is glycine, which only has a hydrogen. Phenylalanine here has a ring system. It's very similar to tyrosine, by the way. The only difference is a hydroxyl. We have cysteine, which does the disulfide bonds, methionine, which does not, proline, in which the ring bends back on itself. The rest of these are pretty standard. Over here, we have some charged or partially charged amino acids, two of which are acidic, so they lose an acidic proton and carry a negative charge, three of which are positive and will gain a proton and carry a positive charge. Histidine is a little bit strange in that behavior. And then you have 5 polar. The polars, they don't have formal charge, but they would interact with water. And remember, All of this has to do with the side chains, not the primary backbone. And so a few tricks to remember. Tyrosine is Y, glutamine is Q, and I've heard this called cutamine, which is kind of a funny way to remember it. Tryptophan is W, and I've heard this pronounced tryptophan in order to help remember that as well. Lysine is K. They literally just ran out of letters. Glutamate, now we would call this glutamate if it has the negative charge, and glutamic acid if it has the proton, but we'll just go with glutamate. The three-letter abbreviation is GLU, the single letter is E, so this forms the word glue. Kind of useful to remember. You are responsible for knowing the three-letter abbreviation and the single-letter abbreviation of all of these amino acids and their classifications. So when in doubt, I guess go with hydrophobic, but then I kind of know that, so maybe you shouldn't go with hydrophobic. Maybe you should go for one of the polar ones if you don't know what it is, but then I would think, well, think of that, so maybe, well, just learn them. You just need to learn these. You should be able to name all 20 amino acids, state which group each one belongs to, i.e. basic, acidic, polar non-charged, or non-polar. You should recognize the three-letter and single-letter abbreviation. I'm not going to hold you responsible for drawing this structure, but I do highly recommend that you be able to recognize the structure. So with that, let's have us a little quick quiz. I'll project a question. You'll have a few seconds to answer it. Okay, so hopefully you got those right. Now, the one about the water-protein interface, with the idea to that one is to keep in mind that the polar and charged amino acids can actually interact with water, whereas the hydrophobic ones probably will not. So if we need a protein to be solubilized, we should expect that amino acid, the key amino acids, to be on the surface so that the water will interact with them. Now that we have the amino acids out of the way, we can look at protein structure. And this is actually very, very simple, yet students seem to have a problem with tertiary and quaternary. So we're going to go over this very, very carefully. The rest of this is very straightforward. Your primary structure is simply the list of amino acids from the amine terminus to the carboxy terminus. That's it. It's nothing more than that. It is defined by the covalent peptide bonds that connect each amino acid to the next, and that's it. So for example, this would be if we had an R1, R2, R3, R4, R5, all we're looking at is the primary sequence. Or similarly, here starting at, for proinsulin, starting at the amine, notice the amine is the NH3 group, we can go phenylalanine, valine, asparagine, glutamine, histidine, leucine, and so on to threonine at the carboxy end. So this single chain here, we're ignoring these disulfide bonds, but this single chain listed as such from amine to carboxy is the primary sequence. The disulfide bonds do not play a role in the primary structure, primary organization. They're very important for higher orders of structure, but not the primary. So in your primary structure, the two things you're really looking out for are the amino terminus. So that's where the amine group is. Notice we have the amine group here. So this is the amine terminus of this sequence. We have the amine groups here. So these are the amine terminus of these sequences, and this one here. And then our other landmark is the carboxy terminus, or the carboxylate. We can see there it is here. That's the ending to the chain, because the chain is always amine to carboxy. Once again, just showing the amine, tyrosine, glycine, phenylalanine, leucine, and here's the carboxy, amine to carboxy primary sequence. Within a sequence, we can have substitutions. Now, these will arise from mutations in the DNA, where we have replacement of one amino acid for another. If the amino acid is of similar polarity, say aspartate, which is negatively charged, and glutamate, which is also negatively charged, we would call that a conservative replacement or conservative substitution. On the other hand, a non-conservative substitution is one in which we change an amino acid of one polarity for one of a different polarity. So let's say threonine, which is polar non-charged, with leucine, which is hydrophobic. That would be a non-conservative substitution. Many proteins and especially enzymes have what are called invariant residues. A residue is just another name for amino acid. An invariant residue is highly conserved. So even across species or across members of a group where we might see mutations hither, thither, and yon, invariant residues probably are not going to change, and if they do, will result in some sort of detriment to the organism. But beyond polarity, something we want to consider, and this is why it does help to know the structures of the amino acids, is volume and surface area. For example, glycine and tryptophan they're basically the same polarity. They're basically hydrophobic, but you can see tryptophan is much larger and bulkier than glycine. This is glycine over here with the two hydrogens. This is tryptophan with the conjugated ring system. And so even though we would see this is a conserved substitution from glycine to tryptophan, we could still see problems because of the size and bulk of the tryptophan residue. So as we move forward into higher levels of protein organization, our next step is secondary, primary being just the sequence. Secondary is driven by hydrogen bonds between the backbone atoms, not the R groups. The R groups are not involved in the creation of these structures, although in some cases in alpha helices the backbone can disrupt it. I'm sorry, not the backbone, the side chains can disrupt it. So local folds, these are just local regions of the peptide that are folded in certain ways, alpha helices, beta sheets, and random coils. So an alpha helix is what it says. It is a spiral. The helix is held together by hydrogen bonds between backbone atoms. Side chains do not play a role, and the side chains project away from the helix. So in these diagrams, we're looking at alpha helices, and these green dots or green spheres, these are the side chains. So we can see they project away from the helix, and just the amine of one end and the hydrogen or the carboxylate of one end and the hydrogen of the amine of the other end, these interact to form the stabilizing hydrogen bond network that keeps the alpha helix together. Now these interactions occur between residues that are 4 away from each other. So if this is residue 1, if I count out 4, 1, 2, 3, 4, this is the residue that will interact with the original residue. Likewise, if we go to here, could be residue 2, let's say, and I count out 1, 2, 3, 4. If I count out the fourth one, that is the one that is interacting. So we have the stagger of hydrogen bonds every 4 residues. Beta sheets, on the other hand, these are not alpha helices. These are comparatively flat, and they adopt a structure that's called either parallel or anti-parallel. The hydrogen bonding between the backbone's atoms, again, is what holds the sheet together, and the side chains project above and below. So it's hard to see here, but these green spheres are projecting away from the screen or back into the screen, and we can see the hydrogen bonds line up within the plane of the screen. If the beta sheet folds back on itself in this way, so we have it going one direction and then it bends around and comes back the other direction, this is anti-parallel. Whereas if the beta sheet is forming by 2 strands in the quote-unquote same direction, that is parallel. So anti-parallel in A and parallel in B. This doesn't have the same restrictions as alpha helices. We can't predict how many residues away the next hydrogen bond is going to be because there's no regularity to the beta sheet as there is with the alpha helix. It's quite a different structure. Beta sheets, each one of these arrows is a beta sheet, and they can adopt additional metastructures of how the beta sheets fold to each other, including things like beta barrels. So the key concept to secondary structure is this is driven by hydrogen bonding, not hydrophobics, not electrostatics, not covalent bonds, not anything like that, just hydrogen bonding. And it's going to be between the amine of one end and the carboxy of another amino acid. Side chains are not involved. Now, the charges and arrangements of the primary structure do determine the secondary structure generated, but this is not a perfect rule. It seems to be more we can infer, but we cannot fully predict. But we do know that substitutions in the primary structure can result in dramatic changes to secondary and higher levels of organization. And that brings us to tertiary structure. Now, the key to tertiary structure is it is within a single peptide chain, not multiple chains. Tertiary refers to one chain, and it is the so-called 3D structure of the protein. This is how the alpha helices and beta sheets fold to each other. So if you think of a secondary structure being how the primary structure folds in a region, then the tertiary structure is how the secondary structures fold to each other. Now here I'm noting the concept of a domain. In general, if we kind of cut out sections of a peptide, they won't fold properly. You cut out a random part of the chain, it won't fold properly without the rest of the chain as context. The exception is called a domain. Domains, if we pull this out, it will still fold correctly into its secondary and tertiary structures, independent of the rest of the polypeptide chain. Now it seems like domains tend to come from specific exons in the mRNA and DNA sequences. A domain usually has a specific purpose. It could be a binding site, could be a structural site, or an interaction site. So let me give you an example. So here we know this is a tertiary protein, and we know that because there's only one peptide chain, and it is folded into a couple of alpha helices and several beta sheets, and how those fold to each other. So we have, represented by this image, primary, that's just the sequence; secondary, that's these beta sheets and alpha helices; and tertiary, and that's how the whole thing folded together. Now we can imagine that maybe if we kind of cut out this alpha helix, it might stay folded properly, and if it did, that would be a domain. And we can kind of imagine this might have a function to close the channel that the beta sheets are making. So if it does, if it has that function, this is likely a domain, which means we could remove this whole segment from the peptide and it would still fold properly. Whereas if we just removed parts of some things, they would not necessarily fold properly. And this brings us to quaternary structure. Now, quaternary structure, this is where students seem to have a problem, is making the jump from tertiary to quaternary. Quaternary is how multiple tertiary structures come together. This means it requires multiple peptide chains, not just one, multiple. Okay? So tertiary structures arise from the folding of alpha helices and beta sheets, how those come together. Secondary structures arise from how the primary sequence folds. Tertiary structures, again, so we'll work our way up. Primary sequence, how it folds into alpha helices and beta sheets, gives the secondary structure. The secondary structure, how those helices and sheets fold to each other, gives you your tertiary structure. Tertiary structures, multiple tertiary structures can come together, and those would be termed a quaternary structure. Now, what holds all this together? The primary structure is held together by covalent bonds. Secondary structure is hydrogen bonds. Tertiary structure is hydrophobic interactions, hydrogen bonds, electrostatics, and even disulfides, which would also be covalent, but that's the only covalent. Quaternary structure can be held together by the same things: hydrogen bonds, hydrophobic interactions, electrostatics, and disulfide bonds. So for example, we'll look at these proteins here. This is probably collagen, and we see three separate polypeptides coming together to form a structure. So this demonstrates quaternary structure. Now note, strangely enough, these do not demonstrate tertiary or secondary structure, because these are not helices and these are not sheets. But it does have quaternary structure because it has multiple peptide chains coming together. This is the proteasome, just to give you an example of something with a ridiculous amount of quaternary structure, and all of these subunits, these are all independent subunits, have their own alpha helices and beta sheets. So this is just an extreme example of tons of quaternary structure. Conformation. So conformation simply refers to the overall final shape of a protein under particular conditions, usually native. You'll hear the terms native conformation, or maybe apo conformation or holo conformation. There's all kinds of these, but it just is saying the shape. This conformation involves electrostatics, van der Waals, hydrogen bonding, hydrophobic forces, and even disulfides. So it's the same sort of stuff, but conformation refers to the overall final shapes. For example, this whole thing is the conformation of this quaternary structure, which lacks secondary structures because there's no alpha helices or beta sheets. Conformation, just to show you, can come from, well, these are mislabeled ionic bonds. These are actually electrostatic bonds, hydrogen bonds, van der Waals interactions, even some disulfides which are not shown. Folding of a protein actually occurs very, very quickly, amazingly enough. So if we look at the dihedral angles of the rotatable bonds within a peptide, there is a minimum of 4 particular angles that are distinct enough that any given rotation can take. So if you have 141 amino acids, that means each one of those, that whole thing has a potential number of configurations of 4 to the 141st power, yet nature finds this conformation almost instantaneously. We as human beings have no means to predict the final fold of a protein from first principles. There's no way for us to do that. Folding, we have a couple of terms here. The native conformation, this is sort of the natural low energy conformation that the protein kind of vibrates around. Denatured, this is if the conformation is lost. Usually you could also refer to this as linearized, though it doesn't have to be completely linear. It just needs to have lost its specific conformation. And folding of proteins is assisted by chaperones. Chaperones are simply scaffolds or enzymes that assist proteins to fold properly. For example, there's an enzyme protein disulfide isomerase that helps coordinate the disulfide bonds. There are heat shock proteins that help keep proteins from unfolding, exposing hydrophobic faces to prevent aggregation. Because if a protein aggregates, it has the potential to fall out of solution, and that would be terrible to a cell. So our key concept in folding is just that each protein usually folds into a small suite of conformations, and it vibrates around these, and these represent the lowest energy states the protein can take. Conformational shifts, on the other hand, so when we want to move a protein shape to a different shape, this can be accomplished, especially for enzymes, by the binding of small molecules, substrates, phosphorylation, and other modifications. So proteins are not static. They are not stationary. They vibrate, they move. Even things that are structural are still moving. Enzymes especially move all the time and can dramatically reconfigure themselves when they bind a substrate. We have 4 sort of general classes of proteins. I'm not going to go into these heavily. They sort of speak for themselves. Globular, which are just what you kind of think of when you think of a protein, just a blob. Fibrous, which is going to be more like collagen. They form rope-like structures, long structures. Plasma lipoproteins, these are just the proteins that are going to carry fatty acids and cholesterol and some other materials throughout the bloodstream. And glycoproteins, which is just about any protein with a sugar label. And now for a disease, and we're going to do an unusual one called the prions. Prions are an infectious substance. Okay, they cause a disease. They are infectious. They are contagious under specific circumstances, but they do not have DNA or RNA. They are not viruses. They do not have a viral coat. They are not cells, so they're not prokaryotes, so they're not bacteria, so they have a plasma membrane. It's just a protein. It is a protein that seems to, in some way, alter the conformation of another related protein. So a couple of diseases related to this are Creutzfeldt-Jakob disease. Symptoms for this are ataxia, dementia, and paralysis. This is almost always fatal. It can be inherited or sporadic, so there are some cases of sporadic mutations, or it can be infectious. And this is also related to Kuru, which can arise from the consumption of brains. So I always like to look out for our zombie brethren, and so please don't eat the brains. Now, when we have a prion disease, there's two hypothetical mechanisms for which this has. Now, the first thing to remember is we have this protein, PRPC, prion protein cellular. This protein you have to have. It appears to be related in neurotransmission or neuronal activity in some way. I'm not very familiar with the protein, but I do know it is a vital protein for us. It exists in a fast equilibrium with an alternative conformation. Same protein, different shape, called PRP scrapie. Now, PRP scrapie comes from the disease scrapie, in which animals that contract this prion end up rubbing up against solid objects until their skin becomes heavily damaged and they can actually severely injure and or kill themselves in this process. So what happens? PRP scrapie will, if there's enough of it around, so we need a critical threshold amount, will polymerize into an insoluble amyloid fiber that is deadly to neuron cells. You need an initiator molecule. It will not happen on its own, but PRPSC is the initiator. So one idea is that PRPSC, as this protein PRPC, briefly transforms into PRPSC and back. If there's enough PRPSC around, either through introduction or mutation, then this will polymerize and fall off solution, which will pull the reaction to scrapie. So that is one mechanism, that we have an ebb and flow issue where scrapie is lost because it falls out of solution, which pulls the PRPC protein to the scrapie conformation. Now, the second hypothesis is that scrapie binds to and modifies PRPC. I'm not sure this hypothesis is holding up any longer, but it does exist. It is out there. We are looking at a protein whose primary structure is beta sheet and who can be extremely insoluble. So here's the idea. We have a nucleus of PRPSC. Either it appears due to mutation or it's introduced in some way, and we have our normal PRPC pool. And as this, remember, this exists in a fast equilibrium, so occasionally a molecule of this becomes PRPSC, at which point it falls out of solution. That pull keeps suddenly pulling tons of PRP into the PRPSC structure, which polymerizes, falls out of solution, kills the brain cells. So here we have normal image of a brain, and here is a prion image of a brain. So I hope this helps you understand the overall basic concepts of the structures of proteins, what forms they adopt, how they're made, the different hierarchies of organization, as well as a very unique disease based entirely on a single protein and not on something complex such as a virus or a bacteria.